A genetic mutation is a change in an organism's DNA that alters the organism's ability to create proteins. This here is our good friend Demetrius Demarcus Bartholomew James III Jr., but we'll call him DJ for short, and he's much heavier than other mice his age. It's always been this way for him, ever since he was young. He's always faced a lot of teasing from his mice peers, but his weight is not his fault. You see, he has a mutation in the MC4R gene located deep inside the nucleuses of his many fat cells. Because of this mutation, DJ's weight has been greatly affected. But before we talk more about DJ, let's dive a little deeper. The MC4R gene codes for MC4R receptors on neurons. These receptors code for the ability to maintain energy homeostasis, regulate the body's food intake, and keep the body's weight in check. But DJ didn't get that privilege. His MC4R gene mutated before birth, and thus was left with the short end of the stick. Some mice develop this mutation in earlier years of their lives, and others inherit these mutations from their parents, but DJ was unfortunately born with it. Here's how it all really works. If we go deep inside the nucleus, which contains all the necessary genetic material of just one of his 3 billion cells, we can find the very start of an intricate process that makes all the proteins within DJ's body. This is known as transcription. During transcription initiation, helicase, a well-known enzyme from the DNA replication process, comes in to unzip the DNA double helix. Another special enzyme known as RNA polymerase helps RNA nucleotides floating around in the nucleus to line up along the template strand of the DNA and follows a base pairing rule. Now, this base pairing rule relates to how nucleotides attach to each other using their nitrogenous bases. There are generally 4 kinds of nitrogenous bases: adenine, thymine, guanine, and cytosine. Adenine pairs with thymine and guanine pairs with cytosine. In transcription, a new nitrogenous base is added, called uracil. Now, when mRNA is being built, it still makes a DNA thymine base pair with adenine, but a DNA adenine base actually pairs with uracil. RNA replaces thymine with uracil as it is better suited for the roles of RNA. Guanine and cytosine will still pair together. As the mRNA is coming to its completion point, the new mRNA molecule peels away from the gene, and the DNA begins to rewind itself. This strand of RNA created from the nucleus is vital as it acts as a carrier for important genetic information. Here's a quick analogy for you. Transcription is like copying a recipe from a cookbook. The cookbook is the DNA containing all the instructions, and transcription is just the process of reading a specific recipe, or in this case, genes. The RNA is like a temporary copy of the recipe, which can be used to create a final dish in the future. In DJ's case, this transcription process, along with many other cellular processes, may occur unnaturally or incorrectly due to the presence of a mutation in his genes. This mutation can affect the efficiency or accuracy of transcription, which could further impact other cellular processes, specifically a process tied together with transcription, known as translation.
Basically, once the mRNA has been created, it leaves the nucleus and makes its way to a ribosome on the rough endoplasmic reticulum. Once there, the mRNA attaches to a ribosome. Because the mRNA is separated into codons of 3 nitrogenous bases, a special type of RNA, known as tRNA, has corresponding bases, known as anticodons. This kind of RNA is floating around, waiting to connect to corresponding mRNA bases in a ribosome. The tRNA also holds a specific amino acid based on the codon it corresponds to, and when attached to the mRNA in the ribosome, releases the amino acid into the ribosome. This begins the amino acid chain, and as more tRNA connects with the mRNA, more amino acids are added to the chain. These amino acids are connected by special peptide bonds between proteins to form a protein chain. rRNA is a special type of RNA that resides within the amino acid to join the amino acids together. You get the idea. The chain only stops once the mRNA codes for a stop codon, a special kind of codon that tells the ribosome to stop adding more amino acids to the polypeptide chain, and after that, the protein is finished. Comparing it to a cookbook once again, translation is the actual addition and application of the ingredients. As you read the recipe, each codon is like the different ingredients. mRNA would be the recipe itself. The anticodons on the tRNA is the chef reading the recipe, and the addition of the amino acids is the physical ingredients being combined together. rRNA is like a whisk. It combines the ingredients together.
Somewhere along the way, either the sperm or egg cell with a genetic mutation of the MC4R gene, when fertilized, replicated over and over and over and over again. And when our friend Demetrius Marcus Bartholomew James III Jr. was born, all of his cells had the mutated gene. But how did it happen in the first place? Well, let's see.
Looking back at the DNA, as we know, DNA is separated into codons of 3. On DJ's 16th codon of 920 codons, codon 16 on the DNA is C, T, T, which codes for mRNA codon of G, A, A. Usually this would be no issue, as it would code for glutamic acid. However, his mutated DNA codes for A, T, T, where A is substituted in as a point mutation instead of C. A point mutation is a genetic mutation that only affects one amino acid in the genetic code. This substitution causes a nonsense mutation, which means when ATT is transcribed into mRNA, it becomes UAA, which codes for a stop codon.
So there it is. That's the story of Demetrius DeMarcus Bartholomew James III Jr. and his life with a genetic mutation, as well as how it came to be.